Um, Hi, everyone. Thanks for joining us today. Welcome to the 102nd MedAI Group Exchange Sessions. And this week we have Chunfeng Lian from XJU TU, China, and he's currently an associate professor of information science and School of Mathematics and Statistics there. His research focuses on machine learning and medical image computing, and he received his PhD in computer science from I'm going to mess up this pronunciation, but this is the university in France. And he was a postdoc with the radiology department at BRIC, UNC Chapel Hill. And he currently serves as an associated editor for 3 international journals, including Medical Physics, IRBM, and Frontiers in Radiology, and has chaired many conferences as well. So thank you so much for joining us today, Chunfeng. Do you have any preferences on how you'd like to take questions? Is it okay if we interrupt you, or would you like them at the end?
I prefer at end, if it's possible for you.
Yeah, that sounds totally fine. So maybe if we all can just hold on to our questions and ask them at the end, that'll be great. And yeah, excited to learn about the meta learning framework from Chunfeng. And without further ado, let me hand it over to you.
Okay, thank you. Okay, so I need to stop my camera for a smooth internet. Sorry for that. Yeah. Okay, thank you, thank Emma for letting me be here, and hello everyone. I'm Chunfeng Lian from Xi'an Jiaotong University. I'm very happy to be here to share with you our recent work on generalized brain tissue segmentation. This is a joint work managed with my PhD student Yongheng Sun and 2 collaborators. One is Dr. Fan Wang from the BME department of our university, and the other main collaborator is Dr. Li Wang from UNC Chapel Hill. So specifically in this work, we propose a dual meta-learning framework for longitudinally generalized brain tissue segmentation across a human lifespan. And we also tried to apply this framework to other cross-domain segmentation tasks. So as a background, we know that brain tissue segmentation is a very fundamental and important task in neuroscience and clinical studies. It is typically formulated as a 4-class segmentation task, that is to assign each voxel to white matter, gray matter, CSF, or the background. However, to construct an accurate tissue segmentation model working for different age groups is not an easy task in practice. This is mainly because the appearance of brain MRI images change dynamically along brain's spatial or temporal developments or degenerations across the lifespan. We can say infancy and sometimes elderly are 2 especially difficult time points. For example, as the figure shown here, we can see that the infant brain changes a lot in the first year after birth, and just isointense around 6 months old makes the differentiation between different tissue types very challenging. In addition, in practice, we usually cannot get too many MR images with the ground truth annotations for a specific age group, which also brings additional challenges to construct a generalized segmentation model. So for brain MR image processing, there are some very famous open source free tools in the community. The first one is the pioneering FreeSurfer, which works well for adult brain. However, some research found that such a general pipeline has relatively worse performance in segmenting infant brain images. Thereafter, an infant-dedicated pipeline called iBeat was developed by the ID Lab from UNC Chapel Hill. It can produce very beautiful infant neural image processing results. But the problem is that the current version of iBeat needs to train an independent segmentation deep networks for different age groups to optimize their performance. which cannot be a good strategy for generalization with very limited data. Therefore, the question now becomes how we can train a universal segmentation model with promising generalization capacity for longitudinally consistent tissue segmentation across the lifespan. In recent years, there are some deep learning works try to do this, especially in recent couple of years. A typical strategy used by them is to design self-supervised learning algorithms to train a common network or feature extractor on longitudinally paired image data, and then fine-tune the network on data from a specific age group for downstream tasks like segmentation. So for example, here is a NeurApes 2022 paper. The authors designed patch level contrastive regularizations to encourage a neuroanatomical consistency across different time points in the representation spaces. Another representative work is by Dr. Paul's group from Stanford. They proposed a self-supervised SSL strategy called local neighborhood embedding to learn subject-level longitudinally consistent feature embeddings, which also needs longitudinal paired training data. We say such SSL method could have some potential limitations, especially from the practical perspective. First of all, we know that longitudinally paired data is very hard to obtain in the neuroimaging community. Also, fine-tuning a pre-trained model could be difficult, especially with limited data. And the commonly used fine-tuning strategy tends to sacrifice the generalization capacity of the pre-trained model for optimizing their performance for a specific age group. Another problem is regarding the patch or subject level regularizations, which may lead to more segmentation errors around the tissue boundaries along brain developments or degenerations. Therefore, the motivation of our work is to answer a question that is it possible to learn efficiently a generalized segmentation model without longitudinally paired data, for example, in a challenging one-shot learning situation? To this end, we designed a dual meta-learning framework for one-shot segmentation across the lifespan. Our DuMeta was implemented under two basic assumptions. The first one is that we assume that in contrast to dynamic appearance changes of brain MRI images along the lifespan, the intertissue differences are relatively stable. And another basic assumption is that we assume a fine-grained delineation of the tissue boundaries requires seamless fusion of such age-agnostic semantics with age-specific details. So according to such basic assumptions, we designed a dual meta-learning framework, which includes two outer loop in the bi-level optimization strategy and a shared inner loop. So in the first bi-level framework, that the inner loop and the first outer loop, we want to learn plug and play or age agnostic feature extractor. And in the second loop, we want to learn lightweight segmentation head with good initialization, because we want to fine-tune such a segmentation head with only one labeled data in the downstream task. So this is the whole pipeline of our DuMeta, and it contains two components. The first one is we want to learn a plug-and-play or age-agnostic feature extractor and a segmentation head with good initialization. And in the meta test stage, we want to fine-tune only the segmentation head with only one label data. So our such a DuMeta framework can be separated as two parts. The first one is to learn the age agnostic feature extractor, and we use the meta feature learning strategy to do this. And the whole story is like this. We want the age agnostic feature extractor can generalize to unseen age group. So we simulate such a situation in meta learning, and we in the loop we fine tune. Sorry, I'm a little sleepy. We fine tune segmentation head on one data set, and to optimize the fine-tuned segmentation head, we in turn optimize the feature extractor with multiple datasets from other age groups. So in meta learning, we construct the implicit function relationships between the parameters in the feature extractor and the segmentation head. And from the equations like this, we can see that actually the generalization is guaranteed by the second order deviations showing in equation 5. And to couple with such a meta feature learning strategy, we designed two regularizations to encourage inter-tissue spatial orthogonality and inter-tissue temporal similarity. And we designed these regularizations different to other regularizations used in similar SSL methods. That is, we use the label information to design these regularizations, but this label information can deduced from only the pseudo labels. That is, we do not need the ground truth labels in meta learning. So another part is the meta initialization learning of the lightweight segmentation head, and this part is very similar to the MAMO method, and it also includes an inner loop and outer loop. So to summarize, we obtain such an algorithm. In this algorithm, we have one shared inner loop, and we have two parallel outer loops. The first outer loops is to train a plug-and-play, a frozen feature extractor that can be generalized across different age groups. And the other outer loop is to learn a well-initialized lightweight task head, which can be fine-tuned with only one label data. So we tested our method on multiple public datasets, which includes WASIS, the IBIS, the ISAC, and the ADNI. And we pre-train or meta-training our model with pseudo label on the WASIS data, the IBIS 20-month-old data, and the IBIS 24 months data. And then we fine-tune our model, only fine-tune the segmentation head with only one labeled data with ground truth for the ISAC benchmark and the ADNI benchmark. And we compared our method with some representative self-supervised learning methods and some meta-learning methods, many the meta initialization methods like MAMO and Reptile. And here are the results we obtained on the ISAC benchmark, and we can see our method lead to better segmentation accuracy in terms of the overlapping metrics and the service metrics. And we need to mention that here we test on the ISAC 2019 benchmark. It only includes the 6-month-old infants, which we did not see during meta-learning. And here are the visualizations of our results. We can see our methods obtain the better boundaries compared to the ground truth. So here we also visualize the cortical surface reconstructed by our segmentation, and which also shows better visualizations of our method. So we also tested our method on the adding dataset for one-shot segmentation of the elderly brain, and the results is showing here, and we can see the results obtained on the adenine is relatively better than that on ISAC, because the first reason is that the elderly brain is relatively more easier to segment than the infant brain, especially in at 6 months old. And another reason is that we already see such elderly brain in the meta training process from the WASIS data. And to show if our method can learn longitudinally consistent feature embeddings by the age-agnostic feature extractors, here we show the 2D visualizations of the top layers of our feature extractors. And we can see, for example, here we show the white matter embeddings of infants and elderly. And we can see from the top 4 layers of our feature extractor, they are smoothly aligned across different resolution and across different age groups, which suggests efficacy of our basic assumption and our meta-learning framework designed on such basic assumptions. So we also conducted some ablation studies to check the efficacy of each key components of our DuMeta, and we can see the DuMeta framework works good, and the inclusion of the class-aware regularization further improve the performance of our DuMeta framework. And another interesting thing that we want to mention is it actually is a trade-off between generalization and specificity. One thing is that the framework we draw, the picture we draw in the first stage is actually a little bit misleading, because we separate the segmentation model as a feature extractor and segmentation head. And the feature extractor actually also includes some layers of the decoders of a segmentation model. So here we can see that the number of layers we left for fine-tuning actually is a very key hyperparameter that we need to optimize. And we found the best results obtained around lefting 3 or 4 upsampling layers in the encoder for fine-tuning. That is, the 3 or 4 upsampling layers are regarded as a feature as a segmentation head. So finally, we also tried to extend this work for other cross-domain segmentation tasks, and we conducted preliminary experiments on one-shot prostate segmentation and used the SAML benchmark. And generally, this benchmark includes the MRI images from 6 sites, and we also train the plug-and-play feature extractor and initialized segmentation head on 5 datasets, and then fine-tune the segmentation head using one label data from the 6 dataset. And we compared our method with some meta-learning methods, including the mammal, the reptile, and also some cross-domain few-shot learning methods that commonly used for this task. And the results show that our DuMeta could also be applied to such a one-shot prostate segmentation task. And the results are shown here. We obtained relatively a better surface dice than other competing methods. So as a conclusion, in this work, we introduce a dual meta-learning framework to learn concurrently an age-agnostic feature extractor, and will initialize the task head. And to train such a network, we design class-aware regularizations to encourage longitudinal consistency, and our DuMeta could need to accurate segmentation models using only one sampled, using only one MRI image with ground truth labels. Finally, we'd like to thank our team of my PhD students and the NSFC for their sponsorship. And here are the link of our paper and code. So thank you for your attention.
Awesome. Thank you so much. This was a really great talk.
Sorry, I'm a little nerdy now. Yeah.
Yeah, that just leaves more time for questions and discussion. So that's totally fine.
Okay.
So I guess before we get started with questions, maybe we can thank our speaker with a round of virtual applause. And yeah, now the floor is open for questions, and
I need to, yeah. So now you can see me?
Yes. Oh, yes, Ramon, go ahead.
Ramon is a... Hi, so great presentation. My question was, so if, let's say for the younger patients, the differences between white matter and gray matter are still kind of consistent. So what could it be present in the images that's causing like regular models to perform poorly on younger brains?
Okay, I'm not sure if I understand your question perfectly. So my answer to this question is like we know from the images we can see. Sorry. We can see that for younger patients, or actually they are not patients, they are the typical developments. So the difference between white matter, gray matter, and CSF, we can see they are hard to visualize, right? And the differences are changes dynamically across the infancy. So, but we assume such differences, such parts of difficult to recognize differences, to differentiate differences are actually from the appearance. And we assume the semantics between these the semantic differences between white matter, gray matter, and CSF are relatively more stable, independent of such appearance changes. So actually, this is our basic idea, and we just want our model to learn such semantic differences, and we assume such semantic differences is actually a guarantee to protect the generalization capacity of a common feature extractor. And we also could say that such an assumption is a relatively weak assumption, and for this end, we may need a downstream task that is the seamlessly fusion of such differences, semantic difference with fine-grained image details to improve the performance. So this is my answer to your question. Yeah.
I might be able to fill in maybe a slightly simpler, problem. The fundamental problem with infant MRIs for the brain is that myelination has not occurred. So except in very limited areas. And so the MRI signal from those areas is much lower in fat and much higher in water. And then there's a continuous process roughly between 0 and 24 months where the myelination becomes substantial enough so that it starts to resemble the mature brain pattern. So basically on a T2 weighted signal, instead of things looking dark, you'll get a large amount of bright signal in a neonatal EEG, and then continuously you'll be, and in a very specific location, location by location, you'll get a maturation of that signal going from light to dark over that 24 hours. So it's a continuous change, and it's location specific. So it's actually somewhat challenging to segment.
Yeah, I totally agree with you, and we can see that the contrasts are changed before 6 months and after 6 months, and this is I think this is a problem you mentioned, and I think a good advantage of the meta-learning is that we know that the segmentation of infant brain is difficult due to the online bination, and the meta-learning framework formulates a link between these infant brain images and elderly or adult brain images, and they just align. This is my conclusion, maybe not correct. They just try to align the differences between white matter, gray matter, and CSF of the infant brain images to the differences of the adult brain images, but just in a nonlinear semantic bit space. So I think this is what meta-learning want to learn from these data. Yeah.
I had a quick question about your meta-learning framework itself. So as you mentioned here, there is a structural difference between the neonatal heads and let's say the adult heads. So the size of the image is also kind of like how do you make sure the resolutions are the same and things like that?
Yeah. It's so in our framework we did not do any registration or spatial sampling to normalize the data to let them have the same spatial size, but we learn the network by two regularizations specifically for the age agnostic feature extractor. These two regularizations is designed with a pseudo label. That is, we used NNUNet, I think, to initially segment the images for the meta training datasets, and then we just use this pseudo label to constrain the semantic differences between different tissues, and also the intra-tissue temporal similarity. That is a longitudinally consistency across infant brains, adult brains, elderly brains. So this is our strategy, and actually, we did not force different images from different age groups to have the same spatial size, and we think we should protect such differences to have a more general or segmentation model. Yeah.
That makes sense. Thank
Mhm.
you.
Mhm.
Oh, and also just a quick follow-up. Is this basically for volumetric segmentation, or is it a 2D segmentation, or will this method kind of generalize to both?
Yeah, so it's actually a 3D segmentation model, but in patch level, we separate each 3D volume as like 200, 128 times 128 times 128, like that volume. Yeah.
Thank you.
Yeah, I had a quick question as well about the data sets you used. So I can see that you have used a wide variety of data sets because you want samples from like different age groups. But is it just the age that makes it difficult for a segmentation model to generalize, or is it, or can there be some dependency on any brain-related, disease or impairment? Did you notice any performance difference, or are there patients or are there samples in your data with some kind of brain-related disease? And did you notice any difference based on that?
Okay. Thank you for your question. It's a very good question. So, yeah, actually, we found the differences between different age groups, and here we selected the ISAC challenge data because we think this is the most difficult the time point for segmentation. The 6-month-old infant brain has iso-intense image, and yeah, for this age group, we have better segmentation results than the other competing methods. But at this age group, the performance of our method is relatively worse than our method for other age groups, like for the elderly data. And we see that here is the results for the ISAC for the 6-month-old infants is like this, and but for the elderly, for the elderly data set, it is like this. So I think the reason, the main reason could be the elderly brain actually it has atrophy, but it is still very clear the appearance between different tissues. And for the elderly brain, it is for the 6-month-old infant brain, it is more difficult. Yeah.
So basically, you see more relationship with age group than any brain-related disease in terms of segmentation performance.
Yeah. Yes, for the elderly data set, we know it has the accelerated brain atrophy that is the AD patients, but we not found clear differences between the performance for NC and AD. Yeah.
No, that's thank you.
Thank you for your presentation. I think I'm looking forward to reading your paper because I'm interested also in how you set up your meta learning framework. But a more practical question, is this something that now we can try out your model with if we have some 6 to 24-month neonatal MRIs we'd like to segment?
Yes, sure. And we release our code, but you may need to retrain the model. But if you want to test with a trained model, we can also send you the trained parameters if you want. Yeah, or we can do it for you. Yeah.
Oh, that sounds great.
On that note, could you tell us the computational efficiency? Like how long does it take for, like, let's say a new training to run, and how many GPUs do you actually need for running your models?
Okay, so thank you. And actually, the experiments were conducted by my PhD student Yongheng, and but we used general GPUs because we are not very rich, and we use 22 GB 3090 the graphics card, and we train the model. The efficiency is not so good because we know the bi-level optimization is relatively more time consuming than general training protocol. And yeah, we train the model, for example, for 1 day, and we can obtain a model with promising performance, I think. Yeah.
Oh, that's great to hear. Thank you. Oh, also maybe just a high level question. Are you thinking of any other like future directions for this work? Do you envision for different data sets or different, like apart from brain tissue segmentation, any other kind of segmentation as well?
Okay, thank you. Thank you for your question. And actually, yes, we have tried to extend this work for other cross-domain segmentation tasks. And at current stage, we just tested on the cross-state one-shot process segmentation. Actually, this is not a very difficult task because it is more like a DG task, right? We have different data sets for the same target, but from different data centers. So we can do it well right now. But we also want to try such kinds of strategies for other more challenging cross-domain tasks, like cross-task tasks, cross targets. For example, we want to obtain a generalized model that can smoothly differentiate between normal brain tissues and lesions. Yeah, right. For like the white matter lesion or the brain tumor gliomas, or we want to obtain a generalized model that can work well across different modalities. These are two main directions that we are studying on. And we also have a similar assumption that is independent of different modalities or different lesions. The differences between abnormality and normality, or the difference between targets, are relatively stable. That is our basic assumption that drive our methodological development. Yeah.
Great, thank you. Do we have any other questions from the audience? Okay, I guess if not, then thank you again for the wonderful talk. And it's also great that the code is open source and then the paper is out. So I think there'll be a lot of interesting follow-up work for other people as well in the call.
Okay, thank you. Thank you for your time.
Yeah, absolutely. And then we'll post this video up mostly by the weekend, and then hope to see everybody next week. So thank you all for joining.
Okay, thank you. So.